Okay, hello everyone. I'm going to get started on the cytology cell lab. So I think what I'm going to do is just run it off the document I have on my thumb drive. That might help let us, well, we still have to share screen differently, but maybe it'll be a little bit faster. Okay, the dog's outside again, so boy. So here is our lab for today. All right. And then I'll have to re-toggle in here to switch to the images. So first, what I want to do is kind of go through and answer some of the questions or fill in some of the blanks, so to speak. So the first exercise is identifying structures and then their functions. So once we have, I'm watching the dog. She's going through a flower pot trying to catch a lizard. She's going to knock over the whole thing. She's a disaster. Anyway, the easiest thing to do in a circumstance like this is to, where all these functions are listed here, is to read the function and then go to the list and try to match it up. It's faster that way. So we're going to start with A, regulates passage of material between the nucleus and the cytoplasm. Well, the structures that really regulate passage of material across it are plasma membranes. And if it's between the nucleus and the interior of the cell, the cytosol, then that would be the nuclear envelope. So we're not going to ask you to point out a particular the structure of the nuclear envelope on the model. And then when we get there, I'll explain why. Said it's not very clear, but you should know the function of the nuclear envelope. So it says here, anything with 2 asterisks cannot be found on the cell model, or not very well, but you do have to know the function. All right, B, transports polypeptide proteins to the Golgi apparatus for modification and packaging. That would be the rough endoplasmic reticulum here. Now you can put the letters in there, or you can synopsize maybe a little bit more what that function is. Most of these are pretty short. And so that's why we give you this, amount of room there for a function. That's about the same size as what the little fill in the blank little sheets would be if we were taking our quizzes face to face. C, the medium which cell reactions occur. That would be the cytosol. Contains digestive enzymes. D, that would be the lysosomes. Where's my little lysosome friend? Oh, down here at the bottom, lysosome. It's going to be our blue jelly bean, I call it. Let's see, E, amino acid assembly. That is the function of ribosomes. F, produces ATP through aerobic respiration. That would be the mitochondria. Let's see, the site of DNA and RNA. Yes, G, the site of DNA, RNA production. That would be the nucleus. H, modifies and packages protein. That would be the Golgi apparatus or Golgi body. Either term is fine. I, contains genetic instructions for the cell. That would be the chromatin. J, synthesizes lipids, cholesterol, steroids, and detoxifies drugs in the liver. That would be the smooth endoplasmic reticulum. K, regulates transport of substances between the cell and the environment. That would be the plasma membrane. L, stores proteins produced by the Golgi apparatus. Those would be vesicles. And it's just not the Golgi apparatus. I mean, any vesicle just stores material, but the lysosomes, well, hold on, let me just check that to make sure we're not calling something this. Hold on, let me go back to L for just a M, anchoring mitotic spindles during mitosis. That would be that would be, where is it, the centrioles. So we have a vesicle, and we have the lysosomes. Yeah, proteins produced by the Golgi apparatus. It's really can be the Golgi apparatus or the smooth endoplasmic reticulum, because those are all vesicles. The reason why it gets a little confusing sometimes, and I think I'm going to change that for next semester to be just stores different products, made by, let's say, the Golgi apparatus. The rough endoplasmic reticulum is in a form of a vesicle. The smooth endoplasmic reticulum has vesicles as well. So it's easy to confuse that with a lysosome. but the lysosome is technically, and we just said that before, is the digestive enzymes. D contains digestive enzymes. So L could be stated a little more clearly or more fully to describe the function of a vesicle. Essentially, a vesicle is just a sac that contains something or stores something for later. However, no one's going to point to that on a model because there isn't one technically on the model. There's technically a lysosome is a type of a vesicle. All right, we're getting TMI. Okay, so let's go to protein synthesis and fill these in, and then we will look at the models. All right, so as I said in lecture, when it comes to protein synthesis, we have certain processes, quote unquote, for you to identify, and cell structures, quote unquote. So the processes are transcription, modification, and packaging, exocytosis, translation, replication. And then the cell structures associated with these processes are protein synthesis, or Golgi apparatus, nucleus, ribosomes, vesicles, backslash, plasma membrane. All right, so those are the possible answers that go in here. All right, so when it comes to DNA, which means that we are replicating, we start with DNA, we end with DNA. That process that's occurring there is replication or DNA replication. That's fine. And the structure in which that occurs is the nucleus. All right, then to have a copy of the DNA in a form that can leave the nucleus, we have to produce RNA. So we have DNA and we end up with RNA. That process is called transcription. This is the way we're going to write down the recipe in a form that can leave the nucleus. And the structure in which it occurs is the nucleus. So two things, the first two things occur in the nucleus. Now we have the RNA that goes out into the cytoplasm, and we have to produce this long chain of amino acids. That production, right, from the RNA is called translation. So now we're going to take what's written here on the RNA and transcribe it and write it into a form that's a linear sequence, right? And so that process is called transcription. And the structure is the ribosome. We said the ribosomes are associated or function in amino acid assembly. Right. Now we have that polypeptide that gets transferred, into the rough endoplasmic reticulum, buds off, goes to the Golgi apparatus thereafter. So now its final form is that from a polypeptide to a functional protein, to work someplace in the cell. All right, that is called modification and packaging. Right. So this process is called modification and packaging, and the structure in which it occurs is the Golgi apparatus, or the Golgi body. Now it buds off there as a vesicle, and now we have this functional protein, and it goes to, the extracellular fluid. I mean, it's not all going to the extracellular fluid, but one of the images that we showed, in the notes was that now that vesicle merges with the plasma membrane and exocytotically releases the contents, that's going to be a primary function, right? Or a primary activity. And I shouldn't say primary. I'm just going to say it's a common activity, not necessarily abundant activity, because making the lysosomes which reside in the cell until it's used, that's a common function as well. But this is an example. Right? So the process is exocytosis, and the structure is vesicles / plasma membrane. All right, this goes rather more quickly. Now we get into how a cell replicates itself. I'm gonna put this on pause just for a second because my dog I just looked over there and she's digging up the garden. Hold on. Get out of there. Oh no. have some water? You want to come in and have some water? Are you thirsty? You know what? What is that there? Your ass in. Go lie down. Go. Go. Okay, so sorry about that. I looked out the window and anyways, I'm home dog sitting by myself, so I don't have any kids to help me. All right, so when the cell needs to replicate itself, right, as they do, um, we have to go through these phases of cell replication. We start with interphase and then we go into, or the next phase is going to be mitosis. Now, mitosis has subphases to it, and thus all this together is considered the cell cycle. So when it comes to replicating itself, we have interphase and the mitosis. The phases of mitosis are prophase, metaphase, anaphase, telophase. So technically you have interphase, then you have mitosis. And then the last activity that needs to be completed is cytokinesis. And cytokinesis means division of the organelles to equally a copy of one cell and then the other. So it's equal division of cytoplasm and its organelles. So you can make essentially a carbon copy of yourself, of your cell. All right, so here it says below number 3 in bold and bold print, mitosis phases are set up. Identify the phases from the slides and models. So you have to identify them on the slides and on the models, as well as put their functions to them, right? Identify centrioles, chromosomes, spindle fibers, cleavage furrow on the models, and identify the phase where cytokinesis is complete. So to make sure we identify those things. All right, so match functions to the phase and apply to models and slides. So first what we're going to do is we're going to identify what functional things, are we going to put in these blanks, that occur during each phase. So we have interphase. During interphase, right, we have here DNA replication and protein synthesis. That goes in the first line, right? DNA replicating protein synthesis. We're making organelles. We're copying our chromosomes so that we have now 2 copies that we can split apart in the middle so that one copy goes to one cell and one to the other. All right. All right. Then we have prophase. So 3 things are occurring in prophase. Chromosomes coil, that's one thing. All right. Centrioles migrate to the poles, opposite poles, and mitotic spindles are produced. That occurs. All right. And then we have, where is the last one in here? The nuclear envelope. The nuclear envelope breaks down. All right. So the nuclear envelope breaks down. Right. So this is what happens. We said that the DNA stays in the nucleus all the time, except when the cell's dividing. So now that we've made an extra copy, all right, we're going to break down the nuclear membrane. We're going to line up the chromosome in the middle and pull them apart. So one copy goes to one end and one goes to the other. Well, we can't line them up all in the center linearly and then in a linear orientation and then pull them apart if we don't break down the membrane. So the membrane has to temporarily disassemble so you can pull apart the chromosomes. Those centrioles, we said that there was just one. Now there's, when the cell needs to replicate, it's got to copy itself. One goes to one end, one goes to the other. They build this kind of a rector set called mitotic spindles that stretch out and grab onto these structures and then pull them apart and pull them to the ends where the centrioles are located. And once you see the pictures and you repeat these functions over and over with each model and each slide, then it's the repetition that starts to make sense. You start to see how you're kind of metamorphosizing from one stage to another and ending up with these products. Okay, so when we see something that represents prophase, you may be asked to give 1, 2, or even 3 different functions that occur during that phase. Right? Metaphase, it's really easy. Chromosomes line up in the middle, right? And that's very, very easy to identify. Right? Then we have anaphase, right? Two things kind of occur, or some people like to consider 2 things. And one is that chromosomes start to get pulled apart. So now that they've been lined up in the center, like in metaphase, now we can start pulling them apart and dragging them to ends of the cell. Right? And also when that happens, we start to develop this pinching in effect called the cleavage furrow in the cell. And so it starts in the much latter portion of anaphase. So chromosomes line up in the middle is going to be metaphase. Cleavage furrow begins, would go here in the anaphase, and chromosomes pull apart, which is over up on the top column here. That goes in there as well. All right, we're almost done. We get to telophase. So now in telophase, now that we've pulled the chromosomes apart, and we start to pinch in and create 2 cells, now we just have to kind of finish that process. So first of all, the 3 things that happened in prophase, the opposite is going to happen now in telophase. So what that means is mitotic spindles let go of those chromosomes. The nuclear membrane reappears and surrounds and recontains all the chromosomes and DNA, so it's not free floating. And the chromosomes now relax. Instead of being nice and dense and tight, which makes them very easy to pull apart, you're going to let them now relax and become all loosey-goosey, like cooked spaghetti, in a pile. Because generally, once the cell completes this mitosis, it essentially goes right back all the way around to interphase again. And it keeps going around and around, depending on how frequently the cell has to divide over and over and over again. So the first 3 things here are going to be the opposite of what's in prophase. And then lastly, we have cytokinesis is complete. That's the 4th thing. So once we re-membrane bound now the nuclei, we've got 2 of them, we've pulled them to the ends, we put them, when the mitotic spindles have let go, we re-membrane bound, the chromosomes get all relaxed, right? Then we have to divide up everything else, make sure everybody gets a Golgi apparatus, mitochondria, a couple lysosomes, so forth and so on. And then we pinch it off in the middle, and now we have 2 little cells. They're called daughter cells. And when you have a daughter cell, it implies that you've gone right back to the beginning and those cells are entering interphase. All right, so we'll look at models of those. Lastly, we have plasma membrane function transport mechanisms. Raw eggs were added to different solutions to observe water movement through a plasma membrane. So, in lecture, we talked about this. I got to sit on my phone. In lecture, we talked about this, what is the effect of the different tonicity of these fluids on a red blood cell? And this is just a way of doing that. We had eggs that sat in certain solutions. And so you would expect that if you have a hypertonic, hypotonic, or isotonic solution, that solution outside of it is going to affect whether water enters the cell, gets drawn out, or has no particular effect whatsoever, right? So all the eggs were added to a vinegar solution. Then afterwards, they were added to one of the 3 solutions listed below, observe the eggs and answer the questions below. So I essentially, even in class, I would have gone over it with you anyway. And then you could go and look at the eggs. You go, oh, no kidding. That's right. That would look like that. Right. And I've identified for you which solutions were what kind of tonicity. That makes it easier. So the first one was a Caro syrup solution. This is a hypertonic solution. So what observation, what was the observation you saw? And it's either going to be the egg shrank, the egg swelled, like we saw with the red blood cells, or they remained unchanged. So if you put a cell in a hypertonic solution, right, it's going to draw the fluid out of it. Water is going to osmose from inside the egg or cell to the external environment. So the observation would be the egg shrank, right? Distilled water, when we put an egg in distilled water, right, that's considered actually a hypotonic solution, right, compared to the egg anyways, right? So So if I put a cell in a hypotonic solution, the water osmosis from the solution into the cell, now the opposite direction of a hypertonic solution. So this egg would swell instead of crenate or shrink. And then egg whites, well, an egg and an egg white, that's an isotonic solution. So the egg remained, egg remained the same size, unaffected. So this is all they're asking you. What effect did caro syrup have on the egg? Caro syrup was a hypertonic solution, so it caused the egg to shrink. Right? Why does this occur? Because water osmosed from inside the cell to outside the cell. What effect did distilled water have on the egg, and why did this occur? It caused the egg to swell. Why? Because water osmosed from from outside the cell or outside the egg to inside the egg. And then we have the egg whites. What effect it had? Neither, because it acted like an isotonic solution. It neither caused the cell or the egg to get larger or shrink. It just stayed the same. So I would understand, since this is really essentially about how red blood cells would be affected in a hypertonic, hypotonic, or isotonic solution. Definitely. I mean, I ask a question or two about it, but that's really about it. I don't show you pictures of eggs. And if I did, I'd say this cell was put in, a hypertonic or hypotonic solution. I think I tell you, if it was caro syrup, I also tell you it's hypertonic. So that then you can anticipate what the observation would be that you would see, or what would be the response of the egg. Okay, so that's quick and easy, filling in those. So let me unshare, and I'm going to go to the web page now. So let me go to Cytology here, and here we have, okay, I'm going to go back to the meeting. Let's share that page right there. Okay, and then I'm just going to change where, no, sorry, wrong one. All right, let me go back into Chrome. If I don't refresh or something. So here we have, let's see if now it'll let me do it. Sometimes you have to like toggle back and forth if you weren't in that. See, and that did it. All right, you have to go in and like do some activity or something, perform some kind of activity in your web page, and then it links it to the share page. Okay, so here we are in Cytology. All right, we had the lab. I just went over this, which is the document, the PDF document, and now we have the images. So we're going to go through the images, and I'm going to try to relate what you have to know from each one. All right, so here we have that composite cell. All right, the model. We have different structures found in here. Here's the nucleus. Here's the nucleoli. You don't have to know the nucleoli in this. You just have to identify those organelles that were in exercise 1 or lesson 1, that didn't have the 2 little asterisks next to it. Everything else are examples of something you should be able to find. All right. So it says structure and function. You're going to look at the different organelles, and their functions, which were essentially given to you in that first exercise. All right. So here we have different organelles here. Now that we've kind of magnified it, I'm just going to tell you that these red jelly beans, this number 8, these are all mitochondria. All right. The big blue ball, is the nucleus. The nuclear membrane is technically some of this that's orangey yellow portion that's intimate with it, but we're not going to ask you to identify that because it's kind of this, is it reference or is it, nuclear membrane? So all this kind of material here, as well as on the side and cut open, they kind of look like caterpillars here, little worms. That's going to be rough endoplasmic reticulum. The little white dots of material in the nucleus is going to be chromatin. Let's see, over here, number 11, the blue sticks here, those are the centrioles. The blue jelly bean here, that's going to be your lysosome. Down here, your brown stacks of sacs, we'll see another image of it, picture of it. That's going to be the Golgi apparatus. for modification of proteins. And then what we don't see here is the clear plastic cup that this comes in, and that's the plasma membrane. All right, so what we tried to do, if you can see them, but we're going to go over them anyway, is that the numbers of on that model that you hopefully can see, the number 1 would be a cytosol. I believe 2 is, no, actually that's not correct, because the RER, well, it could be. All right, that may be correct. Okay, 1 would be cytosol, 2 is plasma membrane, which you're going to see later. 3 is a nucleus, the blue ball. The chromatin is inside. Number 5 is the rough endoplasmic reticulum. And there's a particular picture that shows you just some ribosomes on the surface of a rough ER, and there's little lines emanating from it that merge at a number 6. And that's how you're indicating the ribosomes on the rough ER. And number 11 are your centrioles that produce some mitotic spindles. And they're active in pulling chromosomes apart during cellular replication. Okay, so here we can see some of these. Essentially, the cytosol, here's your number 1, is the light blue watery background. Number 5 here is a rough endoplasmic reticulum, but so are these and so are these. Here's that number 6 I was talking to you about. You have some lines drawn from the ribosomes on the surface of an RER, and therefore that's number 6. Here's number 7. Here's a nice little brown stacks of sacs for Golgi apparatus. Yeah, we're going to go through each one. And here's the plastic cup. All right, so the cell model that you just looked at generally sits in this plastic cup. 2 of them come together, and this is your plasma membrane. Oopsie doodle, I went too far. You know, regulates what goes into and out of the cell, regulates the passage of material across it. Here we have, finally, I said that last week in that lecture when we looked at phospholipids that we'd see something this next week, about a phospholipid bilayer, which is what the plasma membrane is composed of. So here we have it. All right. So here we have a white ball with 2 red tails, and you can't see it really, really well here because the card is in front of it, but you also have up here white balls with red tails. Right. So here we have these phospholipids pointing to each other, and they surround the entire cell circumferentially, as well as surrounds the nucleus and like most of the organelles inside the cell. They are plasma membrane bound as well. All right. So, here we have phospholipids. Each one is a phospholipid. The white ball represents the phosphate group, which is polar covalently bonded of the phospholipid. And the fatty acid tails represent the fatty acids that are non-polar covalently bonded. That's why they point to each other, because they want to be next to each other, because fat likes fat. Right? So the ball would be the hydrophilic end, right, where the phosphate group is, and the fatty acid tails would be hydrophobic. I'm not sure what the card says. You have to find transport lipids, transport lipid molecules, cellular molecules. All right. You can just say that they're constituents of the plasma membrane. That's fine. Let's see. I don't ask you about B, but it's supposed to be a membrane protein. And I've got a call coming in, so hold on just a second. There we go. Let me turn that off. Okay, good. The membrane protein, remember, the proteins, transmembranal proteins, or proteins found to the surface, depending on the size of the channel, if it's a pore versus a channel, because your size, if it's a small passageway, then it just lets ions through and small charged particles. If it's a little larger, then it can let larger molecules get across. So essentially what they're doing, like a membranous transmembrane protein would do if it's acting as a tunnel essentially from one side to another, is to allow either large molecules or polar covalently large charged entities to go from inside the cell to outside the cell, or outside the cell to inside the cell, depending on what direction they're meant to go, or what direction they're meant to be transported. Maybe that's a better way. Okay, so now we have the protein synthesis things, right? We just did that exercise that showed you if I started with DNA and I ended with DNA, you know, what would you have? And then a DNA to RNA. So this is DNA. And let me just look at that. I want to go 2 down because that should be a DNA molecule, but I haven't seen the RNA. So let me, yeah, it says right. Let me just go 1 more. Some of these are hard to see once the pictures. Okay, so clearly a single strand, double strand. Oh, good. I was going to say, I don't know if I can see properly or not. Okay, so we have a DNA molecule and a DNA molecule. And we have what process of protein synthesis. And if this is A and that's B, you know, that A forms B. So you start with DNA, you end with DNA. That process of protein synthesis is called DNA replication or replication. And in what organelle does that occur, you know, or take place? In the nucleus. All right, then we have, if we start with DNA and we end with RNA, then what process of protein synthesis is that? That's transcription. In which organelle would this occur? In the nucleus. Okay. Transcription occurs in the nucleus. All right, then we have, oopsie doodle, sorry, I went too far. We have RNA, and we're ending with a long polypeptide. Are you seeing the pattern here? All right, start with the RNA, end with a polypeptide chain. All right, what process of protein synthesis is that? That is called translation. In which organelle? A ribosome. Okay. Translation and a ribosome. I did say translation before, didn't I? Transcription is when you have DNA and RNA. Translation is RNA to a polypeptide chain. All right. There are multiple models, you know, regarding mitosis. We had what I call the cookie sheet models. And once we get into class and you see one, you'll understand why, because it looks like a great big plastic cookie sheet with different representations on the surface. But we also have what I call cell on stick models, you know, that represent different phases of either interphase of different phases of mitosis. So when it comes to these plastic sheet models, the first 3 things here, number 1, number 2, number 3, you know, represent prophase. Right, so you have prophase, prophase, prophase. You can see a plasma membrane around it in brown. Here you have some cytosol in here. We have the nucleus. Right? We have chromosomes. They look like little X's or bow ties. Right? Here we have centrioles that have now replicated themselves and are migrating to the ends of the cell. And they're going to build mitotic spindles. Right? Okay, so that is, oopsie doodle, prophase, 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 prophase. Interphase is going to be the lower right-hand corner, number 8, where you see the 2 daughter cells. You know, what occurs during, you know, functionally during those phases? Nuclear membrane disappears or breaks down, chromosomes coil or condense, as I said. Centrioles replicate and they travel to opposite ends of the cell and start to build those mitotic spindles, or, yeah, mitotic spindles. There's no other AKA for that. All right, then we go to metaphase. And look, chromosomes lined up in the middle. This is a metaphase number 4. Right. Plasma membrane around to the side, to the periphery. Here we have centrioles. The red lines represent the mitotic spindles. They're like on a Richter set, and they line up the chromosomes in the middle. The blue X's are the chromosomes. That is metaphase. Okay, so we have metaphase, chromosomes line up in the middle. Okay, then we move to number 6. And here, look, we have the chromosomes being pulled apart. It gives you a visual, helps you visually identify what you're looking at. And once you have the function with the phase, then you go, oh, that must be an anaphase. All right, we had the beginning of a little cleavage furrow here. So this is anaphase. Chromosomes are being pulled apart to each respective end. All right, here we have the beginning of what's a cleavage furrow starting to indent in. Right? Anaphase. Right? Anaphase. Cleavage furrow begins, chromosomes pull apart. All right, and here now we have it very exaggerated, don't we? So now we're really pulling these cells apart. That cleavage furrow is really becoming more and more profound. This is the last phase, telophase. So we still have to see interphase, but we had looked at prophase, metaphase, anaphase, telophase, last phase of mitosis. And so what is functionally occurring here? Well, we're going to reestablish the nuclear membrane, kind of comes back or reappears, I guess you could say. And now we have 2 nuclei like this, all right? We have the chromosomes are relaxing, right? They're no longer all condensed like an X. Right? The mitotic spindles have let go of all those chromosomes, right? And now, zip, this is going to pinch off right in the middle, and cytokinesis will be complete, right? That we have divided up the necessary organelles to each cell, and now we have two cells. So I think it says something like that if I had on his card, but we just looked at them. So whatever we have and that we filled, telophase, opposite of prophase, plus cytokinesis is complete. So regardless of sometimes, you know, what some of these little cards say, you know, go with what we've said functionally occurs in the lab exercises, because this is kind of some of these images and things are kind of like, not, I don't want to say vague, like not important, but they don't always include everything that now instructors include. So you can, you know, definitely add to and beef up some of these functions. So they're the 3 things that are opposite of prophase, plus boom, cytokinesis is complete. And now we have 2 daughter cells. So this is what we use, number 8, these 2 daughter cells, to represent interphase. Because once this is complete and have the 2 cells, they essentially go right back into interphase to prophase, metaphase, anaphase, telophase. So the cells can replicate themselves over and over. So this would be interphase. What functionally occurs during that time? Chromosome, a DNA replication and protein synthesis, primarily. We're making organelles, we're doing all that. You know, and most cells spend the most time in interphase. Why? Because you get, you know, it takes a while to copy all the chromosomes and all your DNA, you know, and have duplicate copies of everything, but you have to make duplicate organelles too. And that takes time, because once you start breaking down the nucleus, you have to work fast, you know, and that happens actually rather quickly. Right, so we have interphase and replication of DNA and protein synthesis. Right? Then we have cell on stick models, I call them. Here we have an interphase, prophase, metaphase, chromosomes lined up in the middle, anaphase, chromosomes start to be pulled apart, and telophase. Now you see a really exaggerated cleavage furrow, chromosomes being pulled apart, membrane bounding, letting the chromosomes relax, the mitotic spindles let go of everything. Cytokinesis is complete. Right? So interphase, prophase, metaphase, anaphase, telophase. And you should know now the functions that go with each phase. And that's how questions are generally asked. You know, he just put ITMAP, but interphase, prophase, metaphase, anaphase, telophase from left to right. This shows you some pictures, and your textbook has great pictures too, of what's happening. And this is what, even though our slides don't look quite that good, this is how you can tell the difference between an interphase, a prophase. Interphase and prophase are very similar. However, you generally see when it comes to the nucleus, you can often see a nuclear membrane intact, and the material inside is, and the chromosomes are not as visible as they are in prophase. Why? Because they're all relaxed and you're copying them all, So generally, you don't see it really, really darkened that much, but you do see a membrane. And if we could magnify that, you'd be able to do that. In prophase, the nuclear membrane breaks apart, remember? And the chromosomes condense and coil so when they become dense, they take up stain more, and that stain, now looks much more dark than it did in a relaxed form. So over in this one, if this was in purples, this one happens to be in pink stains, is that it would be, like a medium color purple. Once over here, it would be a very dark, inky purple. All right, and you can see that it's no longer stayed in a nice round mass of a nucleus. They're actually starting to kind of unravel and move, which implies that there's no nuclear membrane keeping it like a nice tight ball. All right, metaphase, chromosomes line up in the middle. Anaphase, chromosomes start to pull apart. See, they're waving goodbye, bye, to their sister chromatid. And here we have telophase. We have still some mitotic spindles between them, but you have a pronounced, an exaggerated cleavage furrow. That's how you're starting to read membrane bound. Some of these mitotic spindles are letting go. Chromosomes are relaxing. No, telophase. Interphase, prophase, metaphase, anaphase, telophase. This is about, this is one that we used to do before, so you can get rid of some of these. I think they just represent those hypertonic, isotonic, and hyper, and isotonic solutions that I talked to you about. I generally don't give you images. If we were in the lab, those eggs in a beaker might be put out, but we're not in the lab. So as far as I know, there's nothing like that on your quiz or the exam. If you find one, and then just let me know. I'll probably just throw it out. Okay. So that's really the lab. We will on Thursday go back over some of these and see what kind of questions you have. And that way, once you've looked at all this, then I can give you examples of questions, and that will make more sense. All right. Okay, so I'm going to stop the share and sign off for now before the dogs wake up and start barking. And yeah, we'll start again on Thursday. We'll go over, maybe now that I gave you all the answers to fill in that sheet, then we'll go back to the images again, and then I'll apply some questions to it. So, all right, then see you Thursday.